Hi everyone, I'm Jamie Batts, your instructor for A&P2. We are continuing on with our lecture on metabolism, energetics. We finished off talking about how most nutrients are converted into a 2 or 3 carbon compound to then enter aerobic metabolism inside of the mitochondria. So we're going to start looking at those processes in more specific detail. Let me switch over to good old screen share mode here, and this is right about where we left off. So talking about the citric acid cycle. Now this cycle goes by several names depending on which source or video you're referring to. Sometimes it's called the citric acid cycle, sometimes it's called the TCA cycle, which stands for tricarboxylic acid. Sometimes it's called the Krebs cycle. So for our book, we're going to refer to it as the citric acid cycle. Overall, the main goal of this process is to remove hydrogen atoms from the organic compounds and transfer them to coenzymes. So the general concept, and I'm going to switch off of screen share mode so you understand this. So remember, we created these 2 or 3 carbon compounds. These smaller molecules have lots of carbon atoms on them. Now here's an important concept. Remember that the energy in molecules is stored in the chemical bonds, right? So if we take a bond from one molecule and we stick it on another molecule, we're transferring energy. This energy transfer can be beneficial to certain reactions because certain molecules are more easily dissociated than others. So in other words, certain molecules are going to lose that hydrogen easier than other molecules, just depending on the chemical properties. So when we take a hydrogen off of one molecule and stick it on another molecule, that is what we call a redox reaction or an oxidation-reduction reaction. We are taking a hydrogen, sticking it on another molecule. When we do that, that new molecule that's created is called, it's oxidized. That's what we call it. Oxidizing is gaining an electron. It's gaining something. In this case, it's gaining a hydrogen. So that new molecule that now has this extra electron, so to speak, this extra energy in the form of this extra hydrogen bond, can now be used to create energy later on in our chemical reactions. So those molecules that are holding these hydrogen atoms, we call them coenzymes. They're going to function in what we call the electron transport chain later on in aerobic metabolism. So the whole process of the citric acid cycle / TCA / Krebs, whatever you want to call it, is to create these coenzymes. We have these 2 carbon compounds, we're pulling off the hydrogens, releasing some carbon dioxide, and we're creating these coenzymes, which are basically placeholders for these hydrogen atoms. Make sense? Placeholders for the hydrogen atoms. So let's break it down into some little steps and get some more terminology here. This whole process is ultimately going to create lots of ATP, and again starts with these 2 and 3 carbon compounds that we created in the very beginning of the reactions. So this happens in the mitochondrial matrix. So those 2 and 3 carbon compounds are created in the cytoplasm. They are then moved to the mitochondria of our aerobic cells. If oxygen is present, this will happen. Those 2 and 3 carbon compounds will move to these cells, to the mitochondria, and a molecule called acetate will be created. This acetate molecule is 2 carbons big, and in the process of taking this pyruvate, which is a 3 carbon molecule, making this 2 carbon molecule acetate, you're going to create 1 NADH and 1 CO2. Guess what? The CO2 is given off, goes to the blood, dissolves in the blood, creates bicarbonate, etc., like we learned in the previous chapters. Or the NADH is not released into the blood. The NADH is this placeholder for hydrogen this is going to go further along in our chemical reactions to give us ATP energy later on. So acetate is this lovely, oh my gosh, my phone is blowing up. I'm sorry. I'm going to turn it down. Acetate is this lovely 2-carbon molecule which binds to a coenzyme A. It's just another molecule. It's kind of like a derivative of a vitamin, and it forms the precursor for this citric acid cycle. So there's this kind of pre-step that takes place in the beginning of the citric acid cycle. You have pyruvate, this 3-carbon compound, converted into a 2-carbon compound, releasing carbon dioxide, right, creating an NADH, combining with acetyl-CoA. Now we have, or combining with coenzyme A, I should say, creating acetyl-CoA. This acetyl-CoA now enters the citric acid cycle. So everything that I just described, creating from that 3-carbon to 2-carbon compound, is this kind of half step or mini step before the real action of the citric acid cycle. So the acetyl group, this CH3CO, this part of the acetyl-CoA, attaches to another 4-carbon molecule. The 4-carbon molecule that it attaches to is oxaloacetic acid. This oxaloacetic acid combines with the acetyl-CoA, and that coenzyme A is now released, and we have citric acid produced. That's the whole point, right? That's why we call it the citric acid cycle. So let me review. Pyruvate, a 3-carbon compound, plus coenzyme A, is going to lose a carbon in the form of carbon dioxide. It's going to be combined with oxaloacetic acid from the citric acid cycle. It's an end product, and this will make sense once you see the whole thing in action. This is going to form a 6-carbon compound we call citric acid. Oxaloacetic acid is a 4-carbon compound. The acetyl-CoA coming in is a 2-carbon compound. Together, it's going to form citric acid, which is a 6-carbon compound. Why does this matter? Because along the way, we're going to be tracing these carbon atoms, because you need to understand that they don't just disappear. They're just converted into different forms. So let's follow this path of citric acid. It's a series of chemical reactions. Like I said, it's mostly redox reactions where these hydrogen atoms are being taken off and put onto a different molecule. They're typically put onto this coenzyme that we call NAD or FAD. When NAD and FAD are oxidized, in other words, when they gain hydrogens, they form NADH and FADH2. They're these little subunits which are going to go on to later form ATP in our chemical reactions. The carbons are always lost as carbon dioxide. The hydrogen atoms are attached to NAD and FAD, and each cycle, each turn of this citric acid cycle is releasing 1 ATP, which isn't that much, considering when you blink your eye, that's like 20,000 ATP. I Googled it. I don't know how accurate that is, but interesting. So these coenzymes, as I mentioned, that are being created from this process are NAD and FAD. NAD is going to carry 1 hydrogen atom from the reduction of citric acid. FAD is going to carry 2 hydrogen atoms from the reduction of citric acid. And NADH and FADH2 are going to move on to this electron transport system, which is going to create lots and lots of ATP. So here's the whole thing in a nutshell. This picture is of the mitochondria. We start with that pyruvate, which was created through glycolysis. Pyruvate loses a carbon through carbon dioxide. It is attached to coenzyme A and combines with oxaloacetic acid at the end of the citric acid cycle. This forms citric acid and begins a brand new cycle. Along the way, and you can just follow this arrow through the diagram, you'll see that more hydrogens are picked off and stuck on to NAD and FAD. Carbons are picked off and released as carbon dioxide. So we're taking this 6-carbon compound citric acid, we're removing 2 carbons along the way, and we're forming that oxaloacetic acid at the end of the cycle, which will combine with acetyl-CoA to start a whole new cycle over and over again. As long as oxygen is present in the cell, this will be occurring. The NAD and FADH2 that is being created from this process goes over into the electron transport system. Here are all of the mini steps, right? Pyruvate reacts with the substrate inside of the mitochondria, releasing carbon dioxide and a hydrogen, which is captured on NAD, forming NADH. It is now a 2-carbon compound called acetate. It combines with coenzyme A to form acetyl-CoA. Acetyl-CoA combines with the end product of the citric acid cycle, which was called oxaloacetic acid. I don't know why it's not on this slide. I guess Pearson wanted to simplify it for you. Either way, forms this 6-carbon compound citric acid. Along the way, oxidation and reduction are occurring. The citric acid is being reduced. NAD and FAD are being oxidized and forming NADH and FADH2. GTP is being formed, which is then converted into ATP. The 2 carbons are lost from citric acid, forming oxaloacetic acid, which will start the whole cycle again. Those carbons are released as carbon dioxide. And our NADH and FADH2s, which are created, go into the electron transport system. So a quick summary for each acetyl-CoA that goes into the citric acid cycle: 5 hydrogen atoms are removed, 2 molecules of carbon dioxide are produced, 2 molecules of water are consumed. Overall, we're a net gain of 1 ATP, and that ATP is actually formed from GTP, which is the actual substrate created in the citric acid cycle. There it all is. There's oxaloacetate. You see all of the substrates. You do not need to know all of the substrates along the way. You do need to know the start and end products, what comes in and what goes out. You need to understand how citric acid is formed, and you need to understand oxaloacetate or oxaloacetic acid, which is that end product that combines with acetyl-CoA to form citric acid. Here's the whole cycle. What I suggest doing is printing out these slides individually and summarizing them along the way. Here you see an oxidation-reduction reaction occurring. NAD is capturing a hydrogen that's coming off of our citric acid, and a carbon is given off. Same thing here. You see that GTP converting into ATP, and another water coming in and oxidizing our substrate there to create another NADH. So this part right here is a part where you can stop the video, go back, review these 3 questions, make sure you understand the citric acid cycle. Sometimes I talk above the book because I think some of the details your book leaves out are important. So we're going to keep going. So the next stop in this process is the electron transport system. And there are 2 things that occur in the electron transport system. One of them is called oxidative phosphorylation. So let's get something, let's stop and understand something. Phosphorylation sounds fancy, but really all it is popping on a phosphate. So you have an ADP molecule and you phosphorylate it and you form ATP, right? Adenosine triphosphate. So oxidative phosphorylation is when you're doing a oxidation-reduction reaction and using that energy, right? Remember, energy is captured from breaking bonds, right? Energy is released from breaking bonds and captured in the form of bonds. When you're taking those reactions where you're breaking and forming bonds and you're actually creating ATP from that energy, right? You're taking the energy from breaking the bonds, typically a hydrogen atom is being broken off, and that energy that was in that bond right there is then captured and used to form ATP from ADP. That's what oxidative phosphorylation is all about. It's using that power of oxidation, that power of popping on or off a hydrogen atom to form ATP from ADP. So there's also something called substrate level phosphorylation, which typically happens everywhere else in the cell. Oxidative phosphorylation is happening in the mitochondria. So just wanted to clarify that little piece for you. Hopefully I didn't confuse you more than I already have, because this is pretty complex. Which is the benefit of having this video, right? You can go back and re-listen to it. You can, put me on repeat if you can stand that, and, listen to the words over and over, read the words over and over. So ATP in the mitochondria, in this sequence of reactions using coenzymes and using oxygen, that's why oxygen has to be present for all this to happen, we will create some ATP, or we will phosphorylate some ADP using that power of oxidation. So we're transferring those electrons. As I've been saying, oxidation is transferring those electrons. Phosphorylation is creating that ATP from ADP. This is 90% of the ATP in our body is produced in this way. So the process of glycolysis, not producing a lot. The process of citric acid cycle, not producing a whole lot, only one, right? Here is where the bulk of our ATP generation is occurring. So let's talk about some key reactions in this electron transport system. It's also sometimes called the respiratory chain, because this is that process of cellular respiration that's occurring, right? We have internal and external respiration exchanging those gases, and the cellular respiration is taking those gases and using them. We're taking the oxygen and we're using that oxygen in this electron transport system to create ATP energy, and then we release carbon dioxide in the process. Oxygen has to be present. It acts as what we call the final electron acceptor. Remember, this whole process is all about transferring electrons, right? We're taking these hydrogens from these big, 6 carbon compounds. We're sticking them on coenzymes, NAD and FAD, and we're sending these coenzymes to the inner membranes of the mitochondria for this electron transport system. What are these hydrogen ions going to do? They're going to create lots and lots of energy. And in order for this to happen, oxygen has to be there to catch these hydrogens. Think about that. If oxygen is there, waiting at the end of the electron transport system to catch, let's say, 2 hydrogens, what do you think the ultimate product of our electron transport system is? Other than ATP, there's one other product that's being released. Oxygen is waiting to catch 2 hydrogens. What molecule is that? Water, right? H2O. So water is created as the very end product, and if oxygen is not there to catch, so to speak, those 2 hydrogens, none of this can happen. The citric acid cycle doesn't happen. This electron transport system doesn't happen. The only thing that happens is glycolysis. So the lack of oxygen is going to stop this electron transport system. How do we get a lack of oxygen? Well, maybe your blood's not pumping enough. Maybe your heart rate hasn't caught up to to the oxygen demand in your tissues. Maybe your tissue perfusion isn't well enough. Maybe your capillaries just aren't used to having all of this blood flowing through, and so they're not very good at exchange, right? That happens, right? That's conditioning. You first start an exercise program and you're very, very sore and you feel the burn, right? Feeling the burn is all of that lactic acid that's building up from anaerobic metabolism, which we're going to talk about in a little bit. As your body gets used to using the oxygen from the blood that's coming through those capillaries, your body's going to be using that oxygen more efficiently to create ATP through this electron transport system. So another way that oxygen can be kind of turned off is by blocking what we call the cytochromes. Cytochromes are these little metal ions which are part of the electron transport system. There are certain toxins like cyanide that can actually turn off this system, turn off the electron transport system. And what does that do? It allows you not to create ATP and not use oxygen efficiently and basically shut down, right? So with no functioning electron transport system, the citric acid cycle is going to stop, and your cells are going to start dying because they don't have the energy that they need. Here is an overall picture of this whole oxidative phosphorylation process. The NAD and FAD come into our electron transport system, which is on the inner membrane of our mitochondria. All of those little purple circles are the cytochromes. These are metal ions like zinc, chromium, right? And these hydrogen ions are basically pumped to the intermembrane space. So NADH and FADH2 are reduced, right? The hydrogens are removed from them, and all of those hydrogens build up in that intramembrane space. It's kind of like water holding back, no, it's kind of like a dam holding back water, right? Think about it. Those hydroelectric power plants are built to hold back water. When one of those gates opens on the dam, water flows through very rapidly. And that is how the power plant generates energy. That rush of water through those little holes is going to turn turbines, which creates electric energy. It's the same concept here. You have this concentration gradient, or a proton gradient, really, because they're hydrogen ions, they're just protons, in this intramembrane space. These protons are stuffed in there. And if you look on the other side of this diagram, you will see a pump there. It's a proton pump. This enzyme, it's an enzyme protein pump, is called ATPase. It's an enzyme that makes ATP. Look at what it does in this image. It's taking the protons, which are stuck in that intermembrane space, pumping them out, right? And it's that same idea of that dam releasing a little bit of water to capture energy. The energy that's captured from allowing some hydrogen ions to pass through is enough to make ATP. This is oxidative phosphorylation. We're using this oxidative energy from these positive ions, these protons, which are in this intermembrane space, pumping them across, allowing them, we're not actually pumping them across, we're just opening the floodgates, letting them flow through, capturing that potential energy and using that to create ATP, right? It's all about potential energy being held back and then being released when we open those channels. Notice what's at the end of this reaction. Those hydrogen ions are pumped through, and what's there waiting for it? Oxygen, right? And water is produced. So the very end products of our oxidative phosphorylation are water and ATP. So here are all of the steps in case I talk too fast or in case you just rather read them than listen to me. These coenzymes, NADH and FADH2, are bringing the hydrogen atoms, which were created from the citric acid cycle, over into the electron transport system. All of these pigment molecules we call cytochromes are embedded in this inner lining of the mitochondria, and these are going to reduce our coenzymes. They're going to take the hydrogens from our NADH and FADH2s. So we're going to release those protons, those hydrogen ions, into that mitochondrial matrix, and the cytochromes are going to pass the electrons down. So the hydrogen atom itself is pulled apart. We have protons going into the intermembrane space and electrons being passed through these series of cytochromes. That released energy is used to pump the hydrogen into that intermembrane space, and those hydrogens diffuse back into the matrix through these ion channels. These ion channels are called ATP synthase. They're special enzymes which create ATP. Oxygen is there waiting in the wings to create water as the final product. Here are those specific cytochromes, B, C, A, and A3. They were all just discovered at different times, which is why they're kind of labeled out of order. But you'll see here we have NADH and FADH2. NADH binds to a product called FMN, and FADH2 binds to coenzyme Q. Either way, the protons are pumped one way, the electrons are pumped the other way through those cytochromes, and at the very end, oxygen and hydrogen come back together to create water. Here it is in a less visual diagram. This is showing you all of the kind of spatial analysis here of these actual particles and where they are. So you see NADH and FADH2 within that mitochondrial matrix, or that inner part of our mitochondria. That's where they were created through the citric acid cycle, and they're going to bind to their subsequent coenzymes through this, inner membrane. those are going to release hydrogen ions or slash protons, it's the same thing, into that intermembrane space. The electrons are going to be passed down through the cytochromes, and at the very end, we have ATP synthase, this enzyme channel waiting there to allow some of those hydrogen ions through. Think of that dam holding back water, allowing some of the water to come through the floodgates, and that energy being captured. That energy is captured, to form ATP. There is a video. This video is not attached to this PowerPoint, but if you go to masteringAMP.com, you can check out this video on the BioFlex. It goes through the same stuff. This is a very conceptual concept. Yes, imagine that. This is something that is very abstract, so it may be helpful to watch a video or two, and even go into YouTube world and type in oxidative phosphorylation animation or citric acid cycle animation, and you'll find lots of them. Just be wary of the source. You would want to watch an instructor or a professor or something from a textbook company, as opposed to little Johnny's 9th grade project, right? So we're going to stop the video here. You can go back and review these 3 questions to check your understanding. And hopefully I didn't talk too fast. And if I did, that's the benefit of a video. Go back and re-listen, re-watch, and have an awesome day. Bye.